Thank you for giving me a chance to present at the MetroFlow meeting. I'm very sorry that I cannot be there in person. I just couldn't do any more travel, but I'm still very happy that I have a chance to give that virtual presentation here. So as you can see, the talk is about high-dimensional cytometry in the spectral era. My name is Florian. I currently work at the Flow Cytometry core facility at ETH Zurich. As usual, I like to give a brief overview of what I'm going to present to you today. So part one is some background and why we actually did what we are doing. Part two is challenges that we faced and the things that we learned on the way, where I want to focus on the things where we feel we really learned something new that could be useful to the community. And that is actually the major part of my presentation. And part three is just some data and controls from the 50 color panel, which I'll keep rather short because this by now is published data. So let's start with part one, background and where we started. So one of the obvious questions is, why do we actually need 50 color flow cytometry? Some people keep telling me there's no need to go any further. We can just run multiple panels and things like that. That might very well be true for certain types of samples. But back then, when we started that project, we were really working on very limited populations, and that is human tissue samples. That's basically our biological question. And if you're in the field of immunology and you just think about all the different types of immune cell subsets that might be relevant to a disease process, T cells, DCs, monocytes, macrophages, you can probably easily name like at least 10 different cell types. And then for each of these cell types, probably at least just from the top of your head, 10 different phenotypes that might or might not matter in a certain disease process. And if you work with human tissue samples, these tissue biopsies, they sometimes can be really, really small as 0.1 grams, and that is all that we have. And the same applies also actually to certain types of blood samples, like, for example, either pediatric blood samples or the ones that are collected by these TASO devices or something alike. In terms of biology, we were mostly interested in understanding the immune response going on inside those human tissues. I won't be talking about that. If you're interested in any of that, you can have a look at the paper that we published a while ago. So basically at the same time, this is when the idea of the 50 color panel started at the end of 2021, which was actually the time at which the Sony ID7000 was installed at the Fred Hutch in Seattle. And I believe at that time, that was one of the first instruments that was out there in the 7 laser configuration, including the deep UV. So naturally, we were super excited and felt like, yeah, we should get going right away. At the time, we had a bunch of existing 28 color panels that were all developed on the FACS Symphony platform that were like really robust, working well. And we thought, well, we can just evaluate some of the newer types of dyes that were coming out at that time, the Nova Fluors, maybe grab some of the old Q dots from the fridge, look at some of the new fire dyes, the new real blue dyes. And then we started adding them based on prior knowledge, some of the published literature, and we thought things will work out fine. So you're all in the field, so you probably know how a nice CD3 and CD19 staining for B and T cells should look like. That's how it should look like. And that's how it looked like when we did our very first panel test. That was like I believe the very first 53 color panel test that we ran back then on the ID7000, where clearly things didn't work very well, which either means we didn't know what we were doing or it was actually more complicated than we thought. Luckily, I think it was in the end more complicated than we thought. So at that time, that was in late 2021, early 2022, the resources available at the time, they did not really help address the issues that we were seeing. So we really went back like entirely to the beginning, back to the drawing board, and we started from scratch in characterizing spectral signatures and fluorochrome behavior of more than 100 different fluorochromes, all of which we ran on the ID7000 and later also on the FACSDiscover S8 when it became available. We looked at fluorochrome brightness. We did a lot of different concatenations. We looked at spreading error, and that's what we thought would be the main problem. And here I want to switch to part two, challenges that we faced and things that we learned on the way. But before I move on to the challenges and the new things, I just want to give a quick reminder of some basics where I feel there's sometimes a misconception, and this is still around, that people feel that when we do spectral unmixing, everything is different just because we do spectral unmixing. And in reality, that's not. I think many of the problems and the concepts that we had when we did conventional flow cytometry are still exactly the same now these days when we do spectral unmixing. So I'm sure I don't need to tell all of you in that audience that usually we try to collect the peak emission of fluorochromes. We do compensation. When we do spectral unmixing, we basically try to collect the entire emission spectrum. And the difference is just the way we mathematically treat the data. And that is here we are solving a square matrix where we have the same number of fluorochromes and the same number of detectors. While over here, when we do spectral unmixing, we are having a rectangle matrix in the sense that we are having more detectors than fluorochromes. So the math is slightly different. However, despite the fact that the math being slightly different, the outcome for the real world problem that we believe is one of the main problems in panel design, spreading error, it's the same. So this is actually some data that is quite old. That was actually a prototype of the spectral symphony from like 6 years ago, I believe by now, or 5 years ago. And here it's really the same single stain run on the very same instrument. And one time it's compensated and one time it's unmixed. And as you can all see, the spreading error that we're getting from that single stain Alexa 700 into either the secondary detector or over here into the secondary fluorochrome in the case of unmixing is more or less the same. Whether it's slightly less or slightly more, that's a topic for another day. But overall, the problem that we know remains the same. So spreading error is present in the raw data, compensated and unmixed data. So we anticipated that this would be our main problem. We just need to deal with spreading error from co-expression patterns and then we'll be fine. In reality, it turns out that this was actually not the case. In the beginning, we also worked a lot with the similarity index, and it turns actually out that the similarity index, while it's very, very useful for evaluating the spectral similarity of fluorochromes, it actually does not help at all in predicting spreading error. And also what we found, and this will be the majority of my talk, that there is a new type of spreading error present in complex panels, meaning anything above like 25, 30, 35 fluorochromes when run on a spectral flow cytometer. Of course, one of the real challenges is also antibody fluorochrome conjugate availability, but as an individual, there's not much you can do about that. We can just be grateful to all the manufacturers which really do an amazing job in making more of these different conjugates availability and working on improving their fluorochromes. So let's talk a little bit about the similarity index. So the similarity index has gotten like really, really popular, and it's a measure of the spectral similarity of two different fluorochromes. However, I think there has been, to some degree at least, this misconception that people actually started using it for panel design. Occasionally, I get to review papers for Cytometry Part A, OMIPS, and the other day I had actually two sets of authors came back during the revision process when I asked about that, and they argued, yeah, but the similarity index, it's a proprietary measure. We just use it and that's it. Well, it's actually a very simple thing. So the similarity index is basically the cosine of the angle of the fluorochrome vector in high dimensional space, or the cosine similarity. That sounds terribly complex, but if you think about it, it's actually really, really simple. So I'm showing you here one example, which is the normalized spectral signature of two different fluorochromes, but you actually cannot see two different fluorochromes because these two green lines, they virtually overlap. It's Alexa Fluor 488 and FITSI. And you can see that there's this minute difference here in this spectral space, but otherwise they are virtually overlapping. So if you think about this now and you basically look at every detector, the signal in every detector as a vector, and you basically sum up all these vectors across the entire detector space, you basically end up with vectors that all overlap, which means you end up with a final vector that basically has an angle of 0 between each. And then if you do the cosine of 0, this is 1. So this would be a similarity index of 1. The other opposite would be if you were to look at two different fluorochromes that have very different normalized spectral signatures, like for example, BV421, which is here in the violet and UV space, and APC Psi7, which is here in the yellow, green, and far red space. And here you can see that their spectral signature is very different, which would mean if you were to sum up all the vectors across all these detectors and then make the angle between two of them, then the angle would be 90 degrees. And if you do the cosine of 90, it means it's 0. And that's a similarity index of 0. And that also tells you that as such, all that the similarity index tells you is basically the overall spectral similarity or dissimilarity of fluorochromes. However, that doesn't mean you can use it as a proxy for panel design. And for this, I want to show you a couple of real-world examples, in this case, measured on an Aurora here at ETH Zurich. So we measured these 5 fluorochromes, BUV661, APC BV480, P5640, and Percy PSI 5.5, and then started looking at them. And if you were to just look at the spectral similarity, you would appreciate that APC and BUV661 are relatively similar, which has to do with the fact that BUV661 is a tandem that includes emission in the same spectral space as APC. So here you have a similarity index of 0.78, which is reasonably high, but nothing to worry about. So what is interesting now, if you look at that fluorochrome pair and you plot a single stain of BUV661 relative to APC, you actually have quite a lot of spreading error, which of course in real world terms for panel design would mean you have a significant loss of resolution here on the BUV661 positive events. Fair enough, that's what you would expect. But turns out when you turn things around and you look at a single stain of APC relatively to BUV661, you can see that there's actually almost no spreading error, which means you almost have no loss of resolution. And as such, this would be a pair if you were to use it that way around, let's say for APC to stain your CD4 and BUV661 to stain a chemokine receptor, in all likelihood, you're going to see like nice separation. But if you were to try to do that the other way around with CD4 on BUV661 and your chemokine receptor on APC, you probably have no chance of resolving any chemokine receptor up here. Having said that, you wouldn't be able to see that from the similarity index because, of course, it's kind of with those 2 fluorochromes, it's just like that's the similarity index they have. But if you were to go back and calculate the good old-fashioned spreading error here, you would have immediately appreciated that here there is a lot of spreading error, and here for this pair there is almost none, and as such could be used for even problematic pairs. The second example is here kind of this in this middle space where you would say that BUV661 and Percy PSI 5.5 and P5640, they are kind of having a low similarity index. But again, if you look at those pairs, again, plotting BUV661 versus either empty Percy PSI 5.5 or P5640, you can see here in this case, you still have quite some spreading error. And in this case, it's low to negligible. And again, if you were to calculate the spreading error here, you would have immediately appreciated that that is the case. And as such, probably made educated choices about how you assign your markers to these fluorochromes. So the gist from that to us was that the similarity index is very, very useful for judging the overall spectral similarity or dissimilarity of fluorochromes, but it doesn't really help you for panel design. And as such, you need to go back to the spillover spreading matrix or use similar matrices like the resolution impact matrix or the, I forgot how it's also called, fluorochrome, loss of fluorochrome stain index. I forgot how Cytometry calls it, that you need to use any of these matrices and only then you can make systematic choices for your panel design. But the other thing that we discovered was actually this new type of spreading error that was very, very puzzling for us in the beginning. So first we kind of thought we were not doing things properly, and then later we actually termed it bloating. And I want to walk you through now why we think that is actually a very relevant thing to look at in the context of spectral panels. So what you can see here is very simple 2 concatenated single stains, a single stain for BV605 and a single stain for BV570. And what you can see is, of course, positives and negatives show quite some spreading error. You actually see this slight tilting of the negatives, which is also a normal phenomenon for spectrally similar fluorochromes. And this is in the context of a 3-color unmixing matrix, which means it's BV605, BV570, and the autofluorescence signature in the unmixing matrix. Turns out when you take the exact same raw data, which are just the 2 concatenated single stains, you don't change anything about the raw data, but you just change the unmixing matrix, meaning you have more spectral signatures in the unmixing matrix. When you apply that more complex unmixing matrix to exactly the same raw data, things look like this. So you can see that the negatives, they show a drastically increased variance or noise or spread, depends on how you want to call it. And I want to emphasize here that these 2 plots, they are exactly on the same scale. There is no change in scales and transformations here. And for clarity, I've put this red box here around, which have the same size, these 2 red boxes. So you can see that there's really a drastically increased spread that affects basically all the events in your measurement. And this is why we initially called this bloating because we didn't really know what's going on here, and we thought it might have something to do with traditional spreading error, but in reality, it's not. And then we were wondering, well, if this effect exists, then we really need to take it into account if we want to continue to do systematic panel design. And for this, we started to look at that in greater detail, and we wondered, how can we quantify it? How can we evaluate it? And we ended up making these kinds of plots that I'm showing you here on the next slide, which is basically a lot of plots. We don't need to look at all of them. Let's just focus on the ones that are in the red squares. So each of these plots is basically one single stain. In this case, it's P phi rate 10. And then the upper row of the plot is that single stain unmixed in the context of a one-color unmixing matrix plus autofluorescence. So two colors, if you want to call it like that. And the lower turquoise plots are the very same raw data, the very same single stain unmixed in the context, in this case, of a 51-color unmixing matrix plus autofluorescence. And as you can appreciate here, there are some fluorochromes, like, for example, that P phi rate 10, where nothing actually changes, or at least nothing obvious changes. While there are some other fluorochromes here, like, for example, PE, or more pronounced even BV510, where you can see a drastic increase in noise, variance, or spread, however you want to call it, of all the cells in your measurement, or the negatives in this case where it's visible. And you can see that this happens in a fluorochrome-specific fashion. And this, of course, always depends on the fluorochrome signatures that you pack into your complex matrix, which at that time were the 51 fluorochromes that we wanted to use for the panel at that time. So the first thing that we concluded here is that this effect seems to be fluorochrome specific. It doesn't affect all fluorochromes equally. And what we also found is that this fluorochromes, this effect really seems to increase on a per fluorochrome basis with the complexity of the unmixing matrix. So here you can see again two single stains, the same type of plots, where the upper row would be the respective single stain unmixed as single color. And down here you can see the same single stain unmixed in the context of a 19-color unmixing matrix, where you can actually see a slight hint of that effect taking place already. Here unmixed in the context of a 35-color unmixing matrix, where the effect becomes more pronounced. Or here we are back to the 51-color unmixing matrix, where the effect is quite drastic, as you've seen it on the previous slide deck. So what we concluded is that this increase in spread is clearly visible and it affects the resolution. But the question remains, besides generating these plots and looking at them, how can we best quantify it with a number and use it for panel design? And what we initially did is we basically just calculated the RSD of unstained cells under these different conditions. And this is basically what this plot here is supposed to show you. So this is actually now the data from the final 50-color panel as it was acquired on the FACS Discover S8, where in blue, you can always see the RSD of the unstained cells in the context of single-color unmixing with that respective fluorochrome. And in orange, you can see the RSD of unstained cells when a full 50-color unmixing matrix is applied to them. And as you can see, there are some fluorochromes where not much is happening, and some fluorochrome neighborhoods, like, for example, here, Berlin Blue 700, or here, the APC Alexa 647 Spark near infrared 685 neighborhood, where you can have up to a 5-fold increase in the RSD of the negatives. So initially, we generated these plots, but then we were wondering whether we can make it yet even more palpable and more easy to use for panel design. And this is when we decided to simply combine it with the stain index. So of course, you're all familiar with the stain index. The stain index is just MFI positive minus negative divided by 2 times RSD of the negatives. And when you do that, you of course get that ranking of fluorochromes, where some fluorochromes on your given instrument are very bright, some are very dim. And this is what you can see here with 2 examples. That's how it would look like for PE size 7 and Brilliant Blue 700. Now I can basically just go and not calculate the stain index as unmixed single stain controls, but you can just unmix your single colors in the context of the full unmixing matrix. And when you do that, you might or might not see an impact on the stain index. And I won't be going now through the entire table that we have here, but just focus on these 2 where we have the red arrows here. What you can see then over here is the raw data. Again, the same plots as I showed you in the previous slide, where the upper row is unmixed as single stain, lower row unmixed as 50-color unmixing, but still the same raw data. And here the same for Brilliant Blue 700. And as becomes evident from the plots, and also here from the stain index, you can see that, for example, PE size 7, in the context of that unmixing matrix that we are having here, is a fluorochrome that is virtually unaffected. So stain index remains the same. While for brilliant blue 700, you would see quite a drastic decrease in the stain index because of that increase in variance or noise in the negatives. And then you can, of course, go and you can just order that adjusted stain index, in this case by the height of the orange bars, and then this would tell you how well you're still able to resolve for a given fluorochrome in the context of the 50 color unmixing matrix with this effect of unmixing spreading error. And this turned out, at least for us, a useful way how we could design the marker assignment for the 50 color panel as we published it in the end. Having explained all of this, I also want to show you something that we found useful, and that is looking beyond the condition number or complexity index as it's commercially known. And that is that the complexity index really tells you something about how well you selected your fluorochromes, but it doesn't necessarily tell you something about where the problems are and how you can fix them. And for this, I want to show you this example. At some point during panel optimization, we were actually considering switching from eFluor 506 to M-Cyan, which is not a great fluorochrome either, but the question is which one actually works better. And if you just look at the condition number or complexity index, by happenstance, which is really funny, these two fluorochrome combinations in the context of like the 50 color panel, they have actually quite a similar complexity index. It's really 40.21, so it's literally the same number. And as you can see also in the neighborhood, the actual similarity indices, they are quite similar, eFluor 506 being here in the middle and M-Cyan being here in the middle. If anything, M-Cyan has actually a higher similarity with BV 480 than eFluor 506. But if you look at the raw data and plot it, you can immediately appreciate that with eFluor 506 and BV 510, you actually have less resolution than you have with M-Cyan. You also have more tilting of the negatives. And more importantly, you actually have less resolution for BV 510, that neighboring fluorochrome, than you do with M-Cyan, which is kind of counterintuitive based on looking at that. Or at least you wouldn't be able to tell that just by looking at the condition number. In turn, if you calculate the unmixing spreading error, or if you calculate the adjusted stain index as I did before, you would be able to capture that effect and realize that with that combination of fluorochromes, you actually get better resolution on the BV510 by swapping from eFluor 506 to M-Cyan. So this is plots that are actually all published in OMIP 102 by now. We've continued some of that work here at ETH to see whether we can use this new metric for selecting fluorochrome combinations in reasonable or even automated manner. And for this, we started generating these types of plots here, which I found actually quite neat, and I'll walk you through that now slowly. So here in the title, you can see the single stain that we are looking at. It's BUV 805. Then here on the y-axis, you can see the fold increase in variance or noise of the negatives. And down here, you can see the number of fluorochrome signatures that went into the unmixing matrix. And each dot here is a random selection of fluorochrome signatures. So for example, here for that particular example, that's an unmixing matrix that consists of 35 fluorochrome signatures chosen from our set of 50 in this case. And it's basically a random selection. And what you can see here is that for BUV 805, basically nothing changes in the RSD of the negatives, no matter whether you unmix it with 2 colors, 10 colors, or 50 fluorochrome signatures. But if you look at BV 510, for example, you can see this increase in noise, increase in variance of the negatives or unmixing spreading error up to 5-fold. And now where things are interesting is basically here. So if you look at that intermediate area, let's say you want to make a 35 color panel, and for whatever reason, resolution in BV 510 matters, then here you can see from this random selection of fluorochromes, you have some combinations which are actually pretty bad. So they generate a lot of unmixing spreading error in the BV 510. Actually, you have a 4-fold increase in variance here, and some which still sit down here, which are pretty good. And that means we are working on either automating that or at least have kind of a shiny app where you can hover over these individual dots and then get the list of fluorochromes that minimizes that lack of resolution, that increase in variance, which leads to a lack of resolution for that fluorochrome. So that's what we are currently working on. And I believe that with standardized instruments, like, for example, the Aurora or the S8 or also the ID7000 with that new normalized settings setup, with these instruments becoming the mainstream, I think one of the important aspects of panel design is really to select the best possible fluorochrome combinations. And that, of course, changes all the time because the manufacturers keep pushing out all these new fluorochromes. And what we found now is that this process can be automated because it turns out that actually the math can be used to predict good and bad combinations. And we are still working on that. So this is a manuscript that we currently have in preparation where we hope that we can basically automate that and really have a tool that would tell everyone, I want to run 35 fluorochromes on the Aurora, or I want to run 40 fluorochromes on the ID7000. And this is my spectral database, which are the best ones. And then you can start working from that in order to minimize that effect of unmixing spreading error that I showed you in the previous couple of slides. So this was kind of the new stuff. And now with the part 3, I just want to show you some data from the actual 50 color panel, but I'll keep that rather short because that's all mostly public, or it's all published by now. So first, here are just the normalized spectral signatures of all the fluorochromes that we ended up using for that panel. And here I wanna emphasize something important, and that is this was work that was started like 2 years ago, and the manufacturers are really working hard in bringing out new fluorochromes. And so this at the time was definitely the optimal set of fluorochromes to achieve a 50 color panel on the S8. But already by now, some of this has changed because new fluorochromes came out. For example, some new real blue dyes came out. Biorad is now coming out with the Star Bright dyes. There are additional fire dyes. So I think this is really work in progress. And this is also why I believe that this automated computational selection of fluorochromes becomes important, because it's completely unrealistic that every lab goes and gets all the new fluorochromes and runs all these measurements on their own instruments. And imagine how great it would be if you can just, from a public database, you can pull the normalized signatures and you can just feed that into the computational pipeline and predict the optimal selection of fluorochromes for your given plexity that you want to run on your cytometer. Having said that, at that time, so that was the best set of fluorochromes we could identify. And these are actually the most problematic pairs per similarity index of the final panel run of a 50 color sample on the Flux Discover S8. And note that this is without any major pre-gating. And you can see, of course, we try to follow the very simple rules of panel design to have these highly similar pairs on mutually exclusive fluorochromes. But you can see even without, it's actually the resolution is pretty nice, and you would even be able to identify probably the double positives here. And that's across the board for all these. So those are the top 5 most similar fluorochrome pairs in that 50 color panel, with the NovaFluor Blue 555 and the Real Blue 545 being the most problematic one. Of course, if you go through the data, I could show you now all the main immune cell subsets. That resolution is nice, but of course we need to compromise somewhere, like for example, the resolution for CD4, CD8, which still is good enough to delineate positives and negatives, but everything else you can see nicely. But what I want to highlight, and what I think is more important, that when you really go down to all these sometimes low expression phenotyping markers that determine the biology of your sample, like for example, here, these 6 phenotyping markers on the T cells like BTLA or CD38 or TIGIT or KLRG1, where the red dotted line always indicates the positivity cutoff. You can see that across all T cell subsets, the mate cells and the NK cells, we have nice resolution across the board. And the same is true here for these phenotyping markers such as CD86 or CD40 or CD11B on the myelate populations, also the small populations like the CDC2s, ones, the different monocyte populations, or here the B cells as a gating control. We can get nice resolution across the board. And again, I want to give here a shout out to all manufacturers that are really working to get these new fluorochromes to the market, because I think they really have a major impact on the biological resolution that we can get. And I want to show you here an example where TIGIT, if you ever stain for TIGIT, you know it's coming up on some effector cells, mostly on the Tregs. And it's kind of, it's a reasonably hard marker to stain. And here you can see the shoulder pulling out. And you could actually say, well, that's okay, I can work with that. But maybe, you know, it could be a little better. And here, this is really an example where we went from one panel iteration. I think this was like, I don't know which panel iteration, but anyhow, we went from here to there. And all that we changed is actually the clone of TIGIT, and we switched from Brilliant Blue 790 to the newer Real Blue 780. And you can see that the resolution that you get is really like day and night, where here you have nice bimodality on the T-Rex, nice bimodality on the effector CD8s. And that's probably true for a lot of fluorochromes. There are new fluorochromes out there that we did not use in OMIP 102, where you can probably even get that improvement for other of your probably relevant markers. I want to wrap up with controls. I mean, as you can imagine, lots of control data are really required to ensure that your panel works as nicely as you think it does. And here I'm just showing you 6 fluorescent -1 controls, 3 centered on the T cells over here. 2 centered on the T cells over here, 2 centered on the T-Rex here in the middle, and 2 centered on the DC population over here. And as you can see, for all of this, we have like basically no spread, no problem in the FMO, very nice resolution in the positive, and it's all clean and nice as it should be. And this brings me to the summary. So what I could hopefully show you here today is that the systematic design of these high-dimensional panels requires new metrics and approaches to select fluorochromes. And I think this is something where we, as a community and as a field, should hopefully come up with the most straightforward way that works in a platform agnostic fashion. So no matter on which platform you work, you can just use public data, hopefully to predict which fluorochromes to use. And afterwards, you just need to work out the co-expression patterns with the traditional ways of like spillover spreading matrix or impacted stain index, which works as well. We believe that the 50 color panel provides a nice and unique approach to interrogate the human immune system in rare human samples with still very well-resolved phenotypic markers. And I also want to emphasize again that this has only been possible because of a strong collaborative effort. The paper is out now, it's OMIP 102, and we really tried to put all information of what we found is useful and what we learned into the supplement. So I think it's a 30-page document, and we really tried to kind of have everything in there that could be useful for the community to use it for your own panel work. And with this, I want to come to the end. I want to highlight that all of this 50 color work was still done in the Privilege Lab at the Fred Hutch. Andrew Konechny is actually the brilliant PhD student who took that over from me and really went through the entire process of doing the final optimization and also doing the final panel runs, which is a lot of work, running like 2 times 50 single stains on. So we always run beads and cells as single stains and run all of those in the S8. You can picture that takes a long time. So thank you to him and Martin for finishing up all that work. I also want to highlight Peter and Aaron, who are 2 wonderful scientists at BD Biosciences with whom we collaborated for that project. I also want to really highlight my colleagues here at the ETH Flow Cytometry Core Facility, Gosha, Annette, Renan, and Irini, with whom I have the pleasure to work now. I want to thank everyone else who helped along the way, and of course, the institutions. And I thank you very much again for giving me the opportunity to present our work at your meeting. And thank you very much for listening, and I look forward to hearing any questions. Thank you very much.
Thank you, Florian. That was fantastic. You can hear me okay?
I can hear you okay. Hello.
Fantastic. So we do have time to take questions from the audience. I'll repeat those. I don't think you'll be able to hear the audience questions so well, so I'll repeat those.
Sounds great. Thank you.
Hi, can you hear me?
A little bit, yes.
So yeah, this is Amritesh. Great presentation, and kudos for the 51 color OMIP. My question is more on the method side. Could you allude to the method of staining, adding antibodies, order of adding antibodies, loss of resolution if you're using cocktails, etc.?
Were you able to hear that? Okay.
Yes, I actually heard it. I think it's about order of staining and like issues of like in which order you stain, if I got it right. So this is a very important question. So first, it turns out we actually couldn't put the 50 antibodies at the correct titers into one mix, so that would have not worked anyhow. But we also definitely saw issues similar to what has been reported in OMIP 69, that when you want to stain TCR gamma delta together with CD3 and a bunch of other T cell markers, that you actually lose resolution on the TCR gamma delta. So this is why we had like 3 sequential stainings, and also we added the right buffers for blocking, which are very important to the correct staining mix. Like, for example, we had the Nova Fluor blocking buffer only in staining mix 2, which had all the Nova Fluor dyes, and these kinds of things. But we list all of that in the supplement of OMIP 102. So in this case, it was actually a 3-step staining, and we list which antibodies went into staining step 1 and which ones went in staining step 2. So, yeah.
So we got Kelly way over here.
What is the for the general criteria? How consistent was that? What was the variance for each floor across the pyramid to have that same criteria?
So Kelly asked, what was the consistency of that floor? So that same floor across experiments. She's got the mic. Maybe you'll be able to hear.
Yeah, consistency in what sense? So hi, Kelly, by the way. Consistency, you mean in terms of resolution that you get, or?
Variance is more what I'm wondering. So say you take BB510, obviously a problem, right? Consistently across each of your experiments, every time you used it, it was a problem for the increase in spreading your agility and mixing. But what was the consistency, the degree of that variance? Was it always the same kind of problem? Was it always just
so you mean the effect? You mean the effect that I showed, that increase in noise, so the unmixing spreading error? Are you referring to that effect? If yes, so if I got that right, if I got that right, so that's basically that is very consistent. So that's really, you can just do the calculation, or you can just run it on multiple days and apply the same unmixing matrix, and that increase in variance, it will always be the same. And that's actually the nice thing, is that you can really mathematically predict it even. So it's really very consistent. It, of course, depends on the unmixing matrix. So if you were to change like a single fluorochrome, as you probably guess, it's about whether you have spectral crowding in a certain area or not. So if you kind of add one fluorochrome that is very close, in this case to BB510, you will drastically increase that effect. Or if you remove one fluorochrome there, you can also drastically decrease that effect. That's what I showed you on those final plots, where I showed you that increase of the effect with the random permutations of fluorochromes. So this is what you see. So one spectral signature removed or added to the unmixing matrix has a drastic effect, but if you keep that the same, it's very consistent. You will always see the same thing.
Okay, so you had 2 instruments that you're also working with. Do you also have an S8? Were you looking at whether or not those same fluorophores were also a problem on other instruments to the same degree?
Yeah, I mean, that's an excellent question. So we actually don't have an S8 here anymore. So at this point in time, I hope we'll get one soon. Having said that, we ran on different instruments, and you will see the same effect on an Aurora, on an ID7000, on an S8, but to varying degrees, because of course the detector array is slightly different, and it depends how on that instrument the spectral signatures crowd in a certain area. So for one instrument, the effect is specific, but it's the same on one Aurora and the other and the next Aurora, and it's the one on one S8 and the next S8 and the next S8. And the nice thing is really, and that I haven't really shown you here, that we found now that you can really use the normalized spectral signatures to predict it. So you can predict it in an instrument-specific fashion, as long as you have somewhere the normalized spectral signatures, which you can even pull from a public database.
Oh, great. Thank you. See, we got one question. We got 6 more minutes. So we got one question on Zoom.
Yeah, we have one question online. Hi, Florian. Is this working, coming through?
Yeah.
Excellent. So the online question is for unmixing the single color turquoise plots with escalating numbers of reference controls. Are the fluorophores that are more similar to cellular or B autofluorescence overrepresented when you get more of the quote unquote bloating that you see? So if the fluors are
Yeah, I mean, this is also an excellent question. So it's, of course, the autofluorescence of the cells is also, of course, modeled as a color. And so you run into issues if you have fluorochromes that are similar to that autofluorescence, which was actually the case for the BV510. Having said that, it's not necessarily the driving factor. The effect that I described becomes a real problem if you want to do multiple autofluorescences, because then you end up with multiple autofluorescences, which actually have very similar signatures, and that leads then to an exaggeration of that effect. But as long as you just have a single autofluorescence, which in this case, it was just PBMCs, it's not really a driver for that effect. It's really kind of the spectral signatures that you either crowd together or don't. That's the driving factor.
Is Anna here? Good to see you.
Hi.
I'm just wondering if you explored or you thought about using different algorithms to unmix, or it was not something that you have explored?
We haven't really explored that in a systematic fashion. So all of this is just done with like standard least square. We haven't really done weighted least square. We've tried it once, but or at least I tried it once. But if you do that, like not within the computational pipeline, but within Flojo, it's actually really tedious. And I haven't seen any major difference though. So it's still, you would see the effect. It might be slightly different, a little more or a little less pronounced, but like nothing. So the difference between the effect being there and not versus the effect with changed with different unmixing algorithms, that was negligible. So changing the unmixing algorithm didn't really change a lot. Having said that, I believe that's a space that will still evolve. Now all the manufacturers are coming out with their own proprietary way of unmixing, and they claim that it's better or not. I think this is something to be seen, whether there's really maybe mathematical tweaks that you can do to the unmixing that will make this drastically better. I don't know. Maybe in the future it will. Right now, I haven't seen that when I tried different unmixing approaches.
Questions?
Yeah, thank you so much, Florian.
Thank you all for listening. It was great discussing, and it's a pity that I'm not there. Hope to see you at another meeting soon. Thanks a lot, everyone.
Thank you.